Welcome to our lecture on bacterial growth requirements. Our learning objectives includes the following. We will define microbial growth, discuss the various nutritional and physical requirements of growth, and we will try to illustrate the bacterial growth curve with explanation of the events occurring in each of the phases. But first, let's define what growth is. In medical dictionaries, it involves an orderly and organized increase in the sum of all components of the organism. The process entails the replication of all cellular structures, organelles, and components. But when we are talking about microbial growth, this is concerned with the increase in the number of cells and not an increase in the size of the organism. What are the nutritional requirements of bacteria? In order for it to grow, this would include carbon, nitrogen, sulfur, phosphorus, inorganic ions, and growth factors. When it comes to carbon, carbon makes up the structural backbone or skeleton of all organic molecules, and based on their carbon source, microorganisms may be classified either as autotrophs or lithotrophs. The second classification would be heterotrophs or organotrophs. Autotrophs bacteria are those that use inorganic compounds or inorganic salts in order to synthesize their food. Their source of energy may be derived from light, and so they could be called photolithotrophs, or their source of energy may be derived from oxidation of inorganic substances, and such as they are referred to as chemolithotrophs. Heterotrophic bacteria used organic compounds like sugars or glucose as their carbon source. Their source of energy may be derived from light, and such as they are called photoorganic drops. But if their energy source is through oxidation of inorganic substances, then we call them chemoorganotrophs. Now, most of the important bacteria are actually chemoorganotrophs. Nitrogen, sulfur, and phosphorus are very essential for a bacteria. To grow, these are essential elements needed for the synthesis of proteins and nucleic acid. For example, protein synthesis is not possible if nitrogen and sulfur is absent. Meanwhile, nitrogen and phosphorus is needed for nucleic acid and adenosine triphosphate synthesis. It is for this reason that when they measure the dry weight of bacterial cell, you'll see that 14 is made up of nitrogen, while 4% is actually composed of sulfur. Inorganic ions needed for bacterial growth include magnesium, calcium, potassium, iron, and trace elements. Magnesium are essential to stabilize ribosomes, cell membranes, and nucleic acids. It also serves as cofactor for enzymatic activity. Calcium, on the other hand, are needed for the synthesis of the cell walls of gram-positive bacteria. It's also a component required for the production of endospores. Potassium is needed for ribosomes' normal functioning and integrity, and it also involved in enzymatic reactions. Meanwhile, iron is found in the cytochrome of bacteria, which is actually part of the electron transport system, or the electron transfer chain. Iron serves as a cofactor for some enzymatic activity. And of course, let us not forget the trace elements. These are components of enzymes and function as cofactors. Some are very important for the maintenance of protein structure. Crude factors are essential to promote the growth and development of the bacterial cell. These include vitamin B complex and amino acids. Just as there is a great deal of diversity in the metabolic properties of bacteria, There is also a great deal of diversity in the type of environments in which different species of bacteria can survive. Microbes are affected in different ways by their physical environment. All living organisms, includes bacteria, require water to survive. However, material species vary in their ability to survive in a dry environment. Although some species will die quickly under dry conditions, others can persist for varying amounts of time. For example, the bacterial endospore is not affected by dry conditions and can germinate to form vegetative cells when it finds itself in moist environment. Other non-spore formers can also persist in the environment for varying amounts of time based on cell properties and amount of glycocalyx that they produce. But generally, bacteria really require water. After all, bacterial cell is composed of water. Furthermore, water is the medium which nutrients are acquired, and it's also the medium where metabolic activities like enzymatic activities happen. Prokaryotes are found in all types of environments. Some microbes are able to adapt in very cold temperatures, whereas others can survive only hot temperature. Each species has a minimum temperature or the lowest temperature that it can grow. They have a maximum temperature, which is the highest temperature for growth, and an optimal temperature, which is the temperature at which it grows best. The ranges of temperature that different types of microbes grow as follow. Bacteria that can thrive in temperatures greater than 40 degrees Celsius are called tempherophils. Those that can thrive between 20 to 40 degrees Celsius are the mesophils, and this is where most medically important bacteria belong. Meanwhile, the cyclophils are those that can survive in 10 to 20 degrees centigrade or even less. So this is a diagram showing the different ranges of growth temperature. So looking at the diagram, you can see that the R group of bacteria is capable of surviving in extremes of temperature and therefore has a wide range of temperature that it can grow. Bacteria has lesser range of temperature growth, and eukarya, or those that have eukaryotic cells, have even shorter range of temperature that is optimum for their growth. Because microbial growth can be affected by temperature, both high and low temperatures can be used to control the growth of microorganisms. One of the most important means controlling bacterial growth is through the use of temperature. For example, low temperatures are primarily bacteriostatic, meaning they inhibit the bacterial growth or they reduce the total number of bacteria. Low temperatures inhibit enzymes activity. So biochemical reactions are slowed down or they could even stop. Because of this, there is a reduction in the rate which the bacteria can metabolize and reproduce. This inhibition of enzymatic activity is usually not permanent, meaning if the temperature increases, the enzymes can function at their normal rate, and the bacteria will resume metabolizing and reproducing. Low temperatures are commonly used to prevent food, for example, from spoiling. Please take note that low temperature cannot be used to sterilize materials. You can only reduce or inhibit bacterial growth, but you can't really kill bacteria in low temperatures. What about high temperature? High temperatures can be bactericidal, meaning they can kill bacteria. When you heat, for example, your inoculating loop in the Bunsen burner flame, this results in sterilization since the bacteria are incinerated. Of course, not all materials can be sterilized by incineration. Both dry heat method and moist heat methods can be used to kill bacteria. Dry heat kills by causing oxidation of cellular molecules and by desiccating or drying the bacteria. Dry heat is commonly used to sterilize materials that could be damaged by moisture. Dry heat methods generally require longer times and higher temperature and are less penetrating than moist heat method. For example, the hot air oven requires 2 hours at 100 to 180 degrees Celsius to sterilize materials. Moist heat, on the other hand, works by denaturation. Denaturating nucleic acids are enzymes in the bacteria. Once these molecules are denatured, they are no longer capable of functioning, and so, even if the temperatures are reduced, the bacteria are incapable of metabolizing or reproducing. Moist heat methods include boiling, pasteurization, and autoclaving. By now, you would be able to translate your knowledge on bacterial temperature requirements in terms of food spoilage prevention as well as food prevention. This brings us to another physical requirement of bacteria, and this is oxygen. Oxygen is an important element because it is needed for cellular respiration. Cellular respiration, if you recall, is the process by which cells produce adenosine triphosphate, which is the energy molecule of the cell. Microbes can be classified based on the oxygen requirements. Those that utilize molecular oxygen for energy production are called aerobes, while those that do not require oxygen are called anaerobes. Some microorganisms are considered strict aerobes, meaning they strictly require oxygen for growth, and in the absence of which it will die. Obligate and aerobes, on the other hand, are microbes that cannot survive in the presence of oxygen. They do not have the enzymes that break down free radicals that are produced in the body. Facultative organisms, on the other hand, survive both in the aerobic and anaerobic conditions. Now, most medically important bacteria are facultative organisms. They are also what you call as microaerophils. These are organisms that can grow at low oxygen environments but will diminished rates. Another group would be the capnophils that required the addition of carbon dioxide to enhance their growth. pH, or the extent of alkalinity of the environment, is another physical requirement for bacterial growth. Alkalophils are bacteria that grow in pH ranging from 8.4 to 9. Meanwhile, most medically important bacteria are neutrophils, surviving at pH of 6.5 to 7.5. Notice that most human tissues would have a pH of 7 to 7.2. Acetophils grow in pH less than 6. Osmotic conditions, or the salt concentration, will also affect bacterial growth. Normal microbial cytoplasmic salt concentration is about 1%, and for a bacterial to grow, the external environment should have roughly the same salt concentration. If there is higher salt concentration, the environment, water will tend to go out of bacteria cell and the cell will shrink, but external concentration of salt is low. Water will come inside the microbial cell. The cell will swell and may even rupture. Organisms that require high salt concentrations are called halophils. Those that require high osmotic pressure for growth are called osmophils. When you talk about bacterial reproduction, this is actually a straightforward process in which each cell splits into two identical new cells. This process is called binary fission. Individual bacterial cells can divide and the bacterial population can double very quickly in as little as 20 minutes in some species. However, bacteria do not continue growing at such a rapid rate indefinitely. This is because factors such as the availability of nutrients and rising toxin levels start to have an effect on bacterial growth. There are different stages of phases of bacterial growth. This would include the lag phase followed by the stationary phase, and the last would be the death or the decline phase. In nature, bacterial growth follows a typical pattern as shown in this figure. The growth curve is comprised of 4 phases. During the lag phase, bacteria are adapting to the environment, are plentiful, and the cell growth in size without dividing. Cell number remains constant. The exponential phase or the log phase marks a big increase in cell number. Maximum growth rate is achieved with constant doubling of the bacterial population. Growth then slows as nutrients become depleted and material base produced build up to toxic concentrations. The bacteria then enters the stationary phase when the number of new cells equals the number of cells dying. The total number of cells in the population remains constant. It is during the stationary phase that some bacterial cells capable of forming an endospores undergo sporulation. Unless nutrients are replenished and waste products are removed, the bacteria progress to the death phase. More cells die than are produced and the number of cells in the population declines. The duration of this phase varies from a few hours to a few days. Of course, any species that were able to form spores or endospores will survive at this point.